que está titulada como Propagación clonal de papa y perspectivas de formación de semillas clonales, a cargo de la doctora Martina Juranic de la Universidad de Wageningen de los Países Bajos. Le agradezco mucho a Martina que nos está acompañando en la mañana de hoy, ya tarde ellos allá en Holanda. Martina es investigadora en mejoramiento genético en la Universidad de Wageningen en los Países Bajos. De aquí en adelante haré referencia como Holanda. Su expertise es la reproducción de plantas con un énfasis en la reproducción asexual por medio de semillas, lo que se denomina apomixis. Martina es oriunda de Croacia, donde ella realizó sus estudios de grado en biología molecular. Después, sus estudios de doctorado los realizó entre una colaboración entre Alemania y Croacia, estudiando mecanismos que regulan el desarrollo sexual en maíz. Después, para su doctorado se fue a Australia para trabajar en apomixis en Zaire. Después, en el 2016, se unió a un proyecto financiado por la Fundación de Bill y Melinda Gates para desarrollar tecnologías para productores agrícolas en África, lo cual es un proyecto que todavía es vigente. Desde hace un año empezó a trabajar en la Universidad de Wageningen, donde continúa trabajando en apomixes y en el desarrollo de técnicas innovadoras de mejoramiento genético. Entonces, con esto le agradecemos mucho a Martina y ya casi arrancamos con...
First of all, I would like to thank for the opportunity to participate in this forum on asexual propagation. Today, I will tell you more about the clonal propagation of potato and clonal seed formation. My talk is divided into 3 parts. First, I'll talk about the importance of potato for the global and Dutch agriculture. Then I'll cover the clonal propagation through tubers and micropropagation of potato. And finally, I'll talk about clonal propagation through seeds, also called apomixis. Potato originates and was first domesticated in South America in the Andes Mountains. Nowadays, the potato is the 4th most important food crop in the world after rice, wheat, and corn. Some other interesting facts about potato. The global potato production exceeds 300 million tons per year, and it's constantly increasing. Potatoes are produced in over 100 countries worldwide, but China is a main producer. Productivity can be variable and it depends on a climate. Potato plants can be very productive, yielding more than 40 tons per hectare within 4 months after planting if grown in North America or Europe. The productivity is lower in Asia, Africa, and Latin America with 10 to 25 tons per hectare. In tropical and subtropical regions of the world, sweet potato and cassava are grown more widely than potato because potato suffers in heat. Potatoes are also used for other purposes than just for food and human consumption. Potato starch is used in the industry. Potato offers high potential and has several benefits. As mentioned on a previous slide, potato plants can be very productive. It is a water-saving plant compared to rice, and it's an important food staple and the #1 vegetable in the world. Apart from a good taste, it's nutritious and it contains more vitamins than other tubers. Potato also generates more employment in the farm economy than any other crops. Netherlands is the largest exporter of seed potatoes in the world, and on the global seed potato market, the Dutch hold dominant position with more than 50% of exported seeds potato. The current export is over 850,000 tons per year. And the reasons for this are that in Netherlands, it is a favorable climate for potato with low virus pressure. The soil in the Netherlands is especially well suited for growing potatoes and allows high productivity. Netherlands has a good certification system and good inspection services. There is an efficient chain approach in production, transport, and trade, and growers are educated and innovative. There is also a good knowledge infrastructure for sharing knowledge and best practices. Also, a broad spectrum of cultivars are available. In my last slide, I mentioned that Netherlands is the largest exporter of seed potatoes in the world. But what are the seed potatoes? Though its name might be deceptive, seed potatoes aren't actually seeds. They are tubers that you can use to grow new potatoes. Similar to any other seed, seed potatoes are potatoes whose purpose is to be planted in the soil to produce more potatoes. And if you have kept a potato a bit too long, like on this picture on the left, potato will develop buds, revealing the potential for vegetative reproduction. And each sprout is a separate new plant. And each plant can produce 50 to 20 new tubers, which will be genetic clones of the mother seed potato, as you can see on the picture on the right. Tubers. Tubers are actually a swollen ends of an underground stem termed stolon. I will just briefly go through major steps of tuber development in potato. It starts with a stolon formation, which is underground stem, and then it is followed by tuber induction, which is characterized by swelling of the stolon tip. And finally, tuber is formed as a starchy storage organ. Tuber formation occurs naturally under short day conditions. Day length and temperature control ensures that tuber formation is completed before winter, when tuber enters dormancy. In spring, it develops sprout, which grows into stems that will eventually emerge from the ground and new plant develops. And then the whole cycle can start again. Potato is responsive to a number of tissue culture techniques, ranging from micropropagation via node cultures to protoplast culture. It is possible to regenerate potato plants from protoplasts. For the production of virus-free plants, it is possible to isolate meristems or induce microtubers in vitro. And also, micropropagation of potato is important for the germplasm storage. So, what kind of research do we do on potato at Wageningen University and Research? We research breeding methods, but we do not perform any breeding. That's what the private sector does. Then plant breeding is famous for mapping in polyploids, and potato is a tetraploid. Then performing QTL analysis and association mapping quality traits, genomics, sequencing, and including gene function analysis. Disease resistance and abiotic stress tolerance are also important topics. And Improving quality is interesting in a way to improve starch content, protein levels, improving frying and baking properties. And we also produce progenitors for special purposes, for example, hybrids of wild species that carry resistance and drought tolerance. Now, I'm moving to the next part of my talk, where I'll be talking about apomixis. But first, let me briefly cover the sexual reproduction. We know that the product of the sexual reproduction in plants are genetically diverse seeds, and this happens through two key processes, meiosis and fertilization. Both of these processes happen within flowers. Meiosis reduces the chromosome number by half and leads to haploid gametes, which are egg and sperm cells. Then, by joining gametes, or egg and sperm cells, through process of fertilization, the diploid embryo is formed, which contains both mother and father genetic material. Later, this embryo will grow into a whole plant, and a new cycle starts again. Asexual reproduction gives rise to a clonal progeny, which are genetically identical to a parent plant. We already mentioned vegetative propagation through tubers, and there are other examples of propagation through bulbs, rhizomes, runners, buds, tillers, and so on. Another type of asexual propagation is apomixis, or asexual formation of seeds. And the term apomixis comes from Greek words apo and mixis, which means away from mixing, and it reflects the fact that meiosis and fertilization are avoided in apomictic plants, and the results will be clonal seeds. Apomixis could be used as a breeding tool to preserve certain traits in continuity. For example, traits like pathogen resistance, early flowering, high yields, drought tolerance, or any other trait. In plant breeding, as shown here, crosses between two different parents may generate a progeny that outperforms both parents and shows superior performance. But due to the features of sexual reproduction, that is, meiosis and fertilization, these traits are lost in the next generation, as shown here in F2 generation. This means that a hybrid seed must be made anew and constantly selected, which costs time and money. And apomixis would provide an elegant way to preserve these traits because progeny would be genetically identical to the mother plant and traits could be fixed in one generation. So without meiosis, without fertilization, there is no trait segregation and the progeny is a clone of a mother plant. However, apomixis is absent in major crop plants, except for a few trees such as citrus and mango. And that is why we study apomixis in natural apomicts. Apomixis is rare and only present in 1% of all species. And among those, 75% of apomicts can be grouped in these three families: grasses, daisies, and rosettes. Below these photos, you can find a list of some apomictic species in each family. Apomixis has evolved independently multiple times, and that's why different modes of apomixis exist. So let's start with sexual reproduction, once again, as a reference. In sexual reproduction, seed is a product of meiosis and fertilization, as we already know. An embryo contains a combination of genetic material from both parents. In citrus, 2 to 10 embryos can develop within a seed, and this type of apomixis is called polyembryony. One embryo, in white here, will have a sexual origin because meiosis and fertilization are functional in citrus. Other embryos are formed asexually from the somatic cells in ovule. The potential problem with the polyembryony can be the nourishment and space within a seed for all embryos that are developing. In two other types of apomixis, called diplospory and apospory, meiosis and fertilization are avoided. In diplospory, a mother cell, which is destined to enter meiosis, in yellow, skips meiosis completely and produces a diploid egg, which will then form embryo without fertilization. And this results in a clonal seed that is a clone of a mother. This type of apomixis occurs in Taraxacum, or in English, dandelion. In the other type, apospory, meiosis occurs, but at the same time, a somatic cell in the ovule, in the pink, reprograms itself into a germline and sexual process stops. The next steps are the same in apospory and diplospory, and both will result in a seed that is a maternal clone. The example of the plant with this type of apomixis is Pilosella on the right. So it would be great to introduce apomixis into crops, and several approaches have already been tried. So apomixis was first tried to be introduced into sexual crops by introgression from wild apomixes. However, this was not successful due to the ploidy, barriers, and sterility of progeny. Next approach is to isolate apomictic genes. A gene, RKD1, in citrus is required for polyembryony, while a gene, baby boom, like identified in apomictic pennisetum, was found to be responsible for a spontaneous embryo development. The search for other genes still continues. The third approach is synthetic or engineered apomixis, where sexual pathway is modified to mimic apomixis. What is needed here is to change meiosis into a mitosis-like division, and this can be achieved by making mutations in 3 genes involved in meiosis. Then it is needed to engineer a haploid induction system to eliminate genome of one parent. And when you combine the two, the result will be the clonal progeny. So this approach was already successfully applied in Arabidopsis and rice. However, the efficiency of synthetic clonal reproduction still needs to be improved, as you can see on this table. In rice, the maximum of 29 clonal seeds was achieved. And just for the comparison, when baby boom-like gene from apomictic pennisetum was transferred into rice, maize, and pearl millet, much higher percentages of spontaneous embryos were achieved. This means that using genes of apomictic origin can give better outcomes. However, this gene was not inducing spontaneous embryogenesis in Arabidopsis. So we still need to find more genes, especially in dicots, which are controlling apomixis. So to summarize my talk, benefits of apomixis are many. First, clonal seeds have an advantage over vegetative propagation because seeds are easier to store and handle. Apomixis would allow a faster and cheaper plant breeding, as we would be able to produce cultivars with fixed traits and durable resistance much quicker. Apomixis would also allow increased reproduction rate because there would be no need for pollination, and consequently, it would contribute towards greater food security. At the end, I would like to thank you for your attention, and I would also like to acknowledge Professor Richard Visser, who is the chair of plant breeding at Wageningen University and Research, for introducing me into the topic of potato asexual propagation. Also, big thanks to my former bosses, Anna Koltunov and Jen Taylor at CSIRO in Australia, where I did most of my apomixis research. Thank you all for attention.